Okay, good morning, everybody. So we're continuing our discussion on gene regulation. So on Tuesday, we discussed prokaryotic regulation with the specific examples of the lac operon and the trip operon, and how those are either repressed or induced depending on the environment. So this is how bacterial cells respond to their environment and are able to survive. And so eukaryotes do the same thing. As you might imagine, their regulation or regulatory mechanisms are quite diverse and very complex. So, and that just comes from the idea that eukaryotes are composed of different cell types that have different needs, function differently, look differently, make different proteins. So this is really, gene regulation is really how we are able to have different cell types that have a diverse array of functions. Okay. So I did find some eukaryotic regulation resources for you. And so this is something to think about, that, you know, gene regulation isn't just about transcribing DNA into RNA. You'll see at the end of today, there are various places within the central dogma where genes are regulated. So genes and their gene products are typically proteins. So we're going to be talking about different places in the central dogma where this stuff may happen. Gene regulation may happen. Okay, and so really it's the big picture is quite simple. You have all these different cells in your body. They, even just this little cartoon here kind of shows that they all look quite different. But they have the same genetic component, or the same genome inside of their nuclei. And so how do you have the same blueprint of information, but you only pick and choose what you're going to use from that blueprint and then actually use it and regulate it and make it into protein products, so that each of these can look different, function different, and interact with different cells. And so really what this is called is, so in different spaces and different times, so spaces meaning different locations in your body, and times meaning different times in your life, so as early as development, and then you go through sexual maturity, and then there is just regular life, adult life, and then you begin aging. So throughout all of these stages of life, different genes are being regulated differently. And so this really is called spatio, so where the genes are regulated, temporal, tempo like time. So it's called spatio-temporal regulation of gene expression. Okay. This is what allows all the cells in our body to have the same DNA in the nucleus, but look and act differently. Okay, and this spatial regulation is absolutely critical for the survivability and sometimes even the viability of the organism. And so I'm just going to show you a couple different examples of why spatiotemporal regulation is important, and what happens when the regulation is not achieved correctly. So we'll look at misregulation of spatiotemporal gene expression. So we'll just start with the fly here. So this is just a cartoon of a Drosophila melanogaster. And so these Drosophila, they are born from, or they are developed from larvae. So at the larval stage, you can't even really tell that it's going to become a fly. It looks more like a little caterpillar, and it kind of does look like that. So what you're seeing here is a schematic of a larval stage of a Drosophila, and in each color, this represents the place, or the space, spatio. This represents the place where certain genes need to be expressed. So here is a strip of DNA. I know it doesn't look like DNA, but just imagine that it's just a line of DNA, double-stranded helix. Okay, this is a chromosome, right? And so in one region of the chromosome, there are these specific genes. The names are not important. It's just to really, the important take home here is that genes that usually function together are usually in the same region of the chromosome. So in one region of the chromosome, you have genes that will eventually function to make the antenna. Okay, and another part of the gene, remember when you have a little slash right here, it means that there's lots of genomic material where we just don't have enough space on this slide. So we're just telling you, yeah, don't forget there's lots of more DNA, but we're not concentrating on that. So in a different region of the chromosome, you have another set of genes, and these are the bithorax genes. And so these genes work together once they're made into their certain proteins. They are responsible for making the back end of the adult fly. Okay. So in the larvae, these antennapedia genes, they're only made in cells in the front part of the body, and they have to be expressed there. So gene transcription of the gene lab, gene transcription or transcription of the gene DFD, SCR, ANTP, those have to be transcribed in these exact regions during this. So this is the larval stage. So this is the time, this is the place. Okay, and that's what has to happen so that when the fly hatches or ecloses from its little cocoon, it actually looks like a fly and all the right body parts are in the right places. Okay, so that's how it should happen. And so we wanted to, before we understood where genes were supposed to be expressed in the fly, we toyed around with genomically inserting genes and making them be expressed in the wrong place. Still the right time, but the wrong place. And so what we ended up doing was we decided, hey, let's see what would happen if we made this gene SCR. It's normally supposed to be in kind of like the beginning of the body. So in the larvae, it's supposed to be right here. These genes are supposed to be transcribed here. Let's make them be transcribed and then translated. Let's make them be transcribed and translated in this region. So the question kind of was right place. Sorry. Down. Right time. Wrong place. Okay, so what would happen? And so what ended up happening is these flies, most of them, I don't know if most of them died, actually, I think they developed, but what you see here, and this is actually the product of that experiment. They took this SCR gene that's normally supposed to be expressed here during the larval stage. They modified a fly so that it is expressed in the head region of the larvae. And lo and behold, what you have are these legs coming out of their face. so it's these legs here are now coming out of the head region. So this definitely shows you that genes need to be expressed in the right place at the right time. So this, if you're looking straight ahead and you're facing a fly, this is what a fly should look like. Okay, is this little proboscis, its nose, its big giant eyes, and it does not have legs coming out of its face. Okay, that's just one example. And this is another example of genes not being turned off at the right time. So this is going to be an example of right place, wrong time. Okay, so this condition is actually called polydactyly. Okay, and this exists in animals with multiple digits. So dogs, cats, humans. And so what ends up happening is early in development, you have genes that are turned on. These genes are cell division genes. And what are you cell dividing? You're cell dividing finger cells. And so what you end up making is a bunch of fingers. And so they have to stem from the base of your hand. And so if you are constantly doing cell division, this is early in embryogenesis, what ends up happening is if you don't turn off those genes, so if you don't regulate that gene and turn it off, the embryo will still keep developing more fingers. So maybe at first glance, this didn't look weird, Or I guess it didn't look, weird is not the right way to describe it, but if it didn't look non-wild type. So at first glance, maybe this looks like a wild type hand, but if you actually count the number of fingers, there are 5 fingers and a thumb. So this person has 6 digits total. And that's because the gene did not get turned off, so at the right time. And so this little kitty here, also you can see a bunch of extra toes. And for some reason, it's really common in cats. I don't know why. And so this is not going to be a detriment to lifestyle or quality of life, but if the person really did want to get the extra digit removed, usually parents will do this right in the first couple months of the baby being born, and it's not a huge deal at all. Okay, and then finally, this is another example. This is the molecular level. So this is something that you can't see from the outside, but you could definitely see it from the inside. So normally when your brain is developing, you have to turn genes on at the right place at the right time. And if you don't turn certain genes on at the right place at the right time, you can experience different effects. Sometimes those effects are detrimental, sometimes they're lethal, sometimes they're no big deal. And so when you're thinking about brain development, neurons don't divide. They come from a pool of neural stem cells. So those neural stem cells are in charge of making all the different neurons of the brain. Okay, that's one population is in charge of making all the different types of neurons, and there's hundreds of different types of neurons. All hundreds of those different types of neurons have their very specific function. They connect to certain things. They're excited by other things. This has to happen properly. Okay, so what happens when you have certain genes turned on at the right or wrong time? So this is just a picture of a mouse brain under a fluorescent microscope. And so what you see on the very far left is basically 1/2 of a mouse brain. So if you took like, I don't know, a knife and cut my face in half, and you're just looking at 1/2 of the brain, and you're looking straight on. So if you were looking straight at the mouse and you're only looking at 1/2 of its brain, this is what it would look like if the mouse was in utero. So day 15, it's an embryo. So usually mice are born at about it's called E for embryonic day, E18 or 19. So this is a few days before the mouse is going to actually be born. And so this is what their brain looks like. And this region right here is called the cortex, which is the part where the mouse is able to perceive its environment and make sense of its environment. Okay, so if you take this little tiny panel and you blow it up or you zoom it in, this is using different antibodies for different proteins to show like, hey, this is the population of neural stem cells. Hey, this is the population of neurons that connect to the elbow. This is a population of neurons that connect to the whatever. Okay, so in this first panel here in FF, you have the antibody that will be specific for a transcription factor. And now we know what transcription factors are, called SOX2. This is not a basal transcription factor, but it's a transcription factor that makes division happen faster. So it's an activator. And so, yes, the cells are in white because usually it's hard to see blue on black. So we just make it a white picture. But every single one of these dots is a neural stem cell. And so this neural stem cell, if it does not express SOX2, it will not properly make the rest of the cortex. And then you can see that if you zoom in, this next little layer is using an antibody called for a protein called TBR2. These are going to be neurons. They're baby neurons, but they're just developing. And so the point here is, we're not going to go through each of these, but the point here is if you look at the merge, so it's all the colors combined, you can see that the cortex is this nice, like layered, looks like one of those rainbow cakes, right? So it's this layered structure. And this layered structure, it has to be in those layers. They have to be made at E15. And if it's not made like that at that period of time, there's no turning back. There's no being late. There's no turning in the assignment in 2 minutes late. It's too bad. The brain will not develop properly. Okay, and so selfless, shameless plug. This is actually my data from my PhD. And so this is my picture, and this is really what I was looking at: is development of the cortex. Are those layers always made properly? Or if you take out certain genes, what happens to that layering? And then what happens to the mouse? And then maybe what happens to humans, because there's a lot of homology between mouse and human brain genes. Okay, so why do things get turned on, turned off? What makes it do that? Yes, there's genes, but why do those genes do that? And it all comes down to what are known as cues, just like cell signaling. So you have extrinsic cues, which are going to be cues from the environment, and that could be the organism's environment, like outside, or it could be the cell environment, like what's happening right outside your liver, right? So it's outside of whatever cells you're talking about, whatever tissues you're talking about. And so we'll start with the organism's environment. So one of the things that can regulate whether genes get turned on or off is temperature. So if you look at this rabbit here, you can clearly see that the nose, the ears, and the paws are a dark color. And so this is a gene that codes for fur pigment. Okay. I think it's eumelanin. Okay. So only when this gene gets transcribed and translated does the protein product is a color pigment, right? So what's happening here is that in this case, the gene is only able to be expressed in low temperatures. Because if you think about it, even right now, this is me, my hands and my feet are always freezing. So that's part of, just having a blood system. It's harder to get blood to the very tips and the extremities. So generally the temperature is colder there. So having a cold temperature is what allows the gene to express this fur pigment. But if the temperature is high, this actually inhibits that gene. Okay? So an external cue inhibiting the gene so that you don't get fur pigment, and that's why the rest of the bunny is white. Okay? So pH. If you look at these flowers down here, the exact same species of flower, the exact same strain, grown in the exact same environment, except for the pH of the soil. So again, it's another color pigment, but pH, so in alkalinic, a high pH is what allows the gene to make a red pigment, and then a lower pH is what allows the gene to make a blue pigment. So pH can affect genes being transcribed or not. And I'm simplifying it here. There's probably a little genetic mechanism that's happening. And so you have also, this is a wild one to me, and it's hard for me to wrap my head around, but you have the availability of mates. Remember what organisms' point is on this earth is to stay alive and to pass their genes down. And so I think this is more prominent in animals besides humans. It's still present in humans, but that evolutionary drive is really, really strong in other animals. And so these are clownfish. And so given the availability of mates in their surrounding environment, they're able to switch sexes so that they can mate and they can pass their genes on. So those are environmental examples of extrinsic cues relative to the organism. Okay, so like your actual environment. And then you can have extrinsic cues relative to the cell itself. So this is when we're like, oh, there's the liver receiving a signal from a different cell type. Okay, those are relative to the cell, but there's still extrinsic cues. So we've seen something like this before, where here is your entire cell. This is the plasma membrane here. And the plasma membrane has these receptors sticking out of it, right? So here's your receptors. And so if a different cell releases a ligand, so these right here are the ligands. And this came from, let's call it an endocrine cell. So the endocrine cell releases the ligands travel through the blood system and reach a different cell type, reach the target cell type. So those are those ligands, and that is an example of an extrinsic cue. And this, to remind you, is called signal transduction. Okay. And the end part of signal transduction is that a transcription factor, an active transcription factor, will either activate or repress transcription. And so usually signal transduction has a bunch to do with things getting phosphorylated, and you can see that that is also the case here. So all these things, phosphorylates, activates, okay, fine. And this right here is a transcription factor that is active. What does it do? It translocates into the nucleus. It finds the target gene, and now we know where it would bind in the target gene. It's going to bind the promoter. Okay, so here's your AR. It binds the promoter, recruits other transcription factors too. What else does this cartoon show you that you now have seen in previous lectures? This is your DNA being wrapped around histones. So these gray columns are your nucleosomes, and here's your DNA being wrapped around it. So in this situation, the promoter was available, was accessible. So this extra transcription factor can bind, and then it makes genes, so it makes them into transcripts for growth or survival or whatever, okay? And so these are the mRNA versions, the mRNA version of these genes, and then these mRNAs leave the nucleus, head to the ribosome, make a protein, and then the cell reacts. So the gene has been turned on. Okay, that is regulation of gene expression. Okay, so those are your extrinsic cues. Just an example of an extrinsic cue. And also putting together stuff that we've talked about in the past. Okay, and so as opposed to extrinsic cues, you have stuff that's happening inside of the cell, and those are called your intrinsic cues. So this is within the same cell. Okay. So if you have a severe increase or a decrease in concentrations of important molecules, like glucose, or like ATP, or like ADP, or any of these molecules that, you know, really just allow the cell to function, that is going to, that's going to regulate whether genes are being made or not. So if you have a high level of ATP, then you're probably going to stop, this is just one example, stop or inhibit genes that code for cell respiration. Genes that code for cell respiration enzymes, right? Because if you have a bunch of ATP, then you better stop making cell respiration enzymes because we don't actually need them right now. And then the opposite is true for ADP, right? And so that happens within the cell itself. That's an intrinsic cue. And you can come up with a lot of different examples of this. So I just want to mention a couple. And so damage is another thing that happens inside of the cell, and the cell recognizes the damage. You might say, yeah, but didn't the damage happen because of UV rays? Yeah, but the cell doesn't recognize UV rays. The cell recognized the effect of UV rays, so the cell recognizes damage. Damage is happening in the cell. So one example here is, you know, your DNA damage. Okay, and so if DNA damage has occurred, then you're going to turn on the repair machinery. But then you better recognize when the damage is fixed so you can turn off the repair machinery. Okay, so balance of on and off has to happen all the time. They're switches, and they're actually called molecular switches. When you switch the genes on for repair, then you need to activate the molecular switch so that it turns off. And that's just DNA. But if any of the organelle damage, if any of them are damaged beyond repair, then you probably elicit phagocytic genes. right? So make sure we make more phagocytes, or make sure we package that debris into vesicles, or whatever that may be. Or if you can fix it, fix it. Okay? So just things to think about, because those genes have to be turned on only when the organelle is damaged, because if they're always on, they don't know the difference between damaged organelles and not. So if they're being made, they're just going to find the mitochondria and start gobbling it up, or finding the, broken ribosome and tearing it apart, whatever it may be. Okay, so regulation doesn't just happen at the level of transcription. So it's not just about turning transcription on or off. There's different places beyond that where we can say genes are regulated. So these asterisks here indicate regions that can be modified. So here's your typical central dogma of molecular biology, where DNA is the template to make, so this is eukaryotes, DNA is the template to make your pre-mRNA. The pre-mRNA is processed, so it becomes the mature transcript. The mature transcript leaves the nucleus and heads to the ribosome, so that the ribosome and tRNAs can make the amino acid chain. Then this amino acid primary structured peptide gets folded and modified to become a functional protein. So at all of these places, can we either stop this process from proceeding, or we can make it happen a little bit faster. Okay. And then what are these other lines that I have here is to remind you that the central dogma of molecular biology is not this linear process. Okay, so DNA, oh, I don't know why my little arrow heads, what happened here? Okay. So yes, you can have this modified protein, but it's these proteins that are the replication machinery. Some of these proteins become transcriptional machinery. Some of these proteins allow the mRNA to be translated. Some of these proteins help the amino acid chain be modified, like kinases phosphorylate amino acid chains, right? So it's this big circular network of processes that all have to happen correctly. So more steps to do right, more chances to mess up. Okay, which means you should have a bigger appreciation for your cell's ability to not make mistakes. I think that's really great. So essentially, we're just going to go through each of these throughout the rest of the lecture. So before I get started with all that stuff, I see some stuff in the chat, so let me just make sure no one has any questions. Okay, all right, cool. All right, so any questions? Okay. All right, so let's go ahead and get started. So one place obviously is regulating transcription, and this can happen in two different mechanisms. You can either regulate transcription by whether or not the promoter is accessible, or you can regulate transcription via these extra transcription factors. So you can regulate the rate, or you can regulate whether or not the promoter is accessible. And so this, maybe you haven't seen this picture before, but you definitely have learned these concepts before, where certain amino acids of histones, remember the lysines, they can be methylated or they can be acetylated. So here's your methyl groups in green. And then you have acetyl groups in orange hexagons, okay? And so remember that if a methyl group is attached to histone proteins, this tends to make the DNA wrap tightly around the histone. So the nucleosomes are squished and tighter together. So that means the DNA, more specifically, the promoter is inaccessible. Inaccessible. And you can see the little gene right here. So here's the whole gene, and maybe let's choose a different color, blue. Maybe the promoter's like, no, terrible color. No? Maybe the promoter's like right in that region. And the transcription factors can't get there. RNA polymerase can't fit in there. So the gene is not transcribed. So we call this, when the histone is methylated, we call this phenomenon, so this is chromatin, right? And so why am I spacing? This is crazy right now. Sorry, I just had like a lapse of my brain. This is called heterochromatin. Okay, stuff that I studied for years just left my brain. Okay, heterochromatin, that's the name of it when the chromatin is so tightly wound. Okay, the flip side of that is called euchromatin. Euchromatin is something needs to remove these methyl groups, and then instead you have these acetyltransferases that are going to put acetyl groups onto the lysines, and this forces the DNA or promotes the DNA to wind loosely around the histones, thus making the promoter accessible, which means transcription factors can come and bind the promoter, RNA polymerase can come and start transcribing this region. So euchromatin is associated with acetylation of not only histones, but even the DNA itself can acquire what these are called marks. So essentially what we're talking about here is versions of epigenetics, and I'm sure you guys have heard this term before. So epigenetics means you're not actually changing the sequence of the DNA, but you are either adding functional groups or taking functional groups away that make transcription either, that either allow for transcription or not. So this is not mutation. So we're not changing the nucleotide sequence. It is either adding functional groups or removing them so that the gene region is accessible or not. Okay. Affects accessibility. Promoter, and you might have seen it in cartoons or maybe previous slides, but scientists for some reason like to draw the symbol for promoter as an arrow indicating this is where transcription starts. Okay, great. So just remember this is methyl groups, and this is usually lysine, arginine, and then the N-terminus of histones. Okay. And then acetyl groups are typically going to be modified and added, not modified, acetyl groups are typically going to be added to lysines, again, of histones. But the DNA itself can also be methylated or acetylated. These are called marks. So this is a mark, and we call them methyl marks, acetyl marks. Okay, so that's your promoter accessibility, and it all comes down to your chromatin assembly. Is it a chromatin heterochromatin or is it euchromatin? So you have transcriptional regulation. So this is actually making transcription happen faster than the normal level. So don't forget, you have these things called basal transcription factors. Those are the ones that recruit RNA. Those ones are always going, if the promoter is accessible, those ones are always going to be there. For every single gene in the eukaryotic genome, you have a set of basal transcription factors. Let's call them basal TFs. These are the ones that recruit RNA polymerase. That never changes. That's always going to happen. So you basically always have a very low level or very basal level, low basal level of transcription, unless something's blocking the RNA from polymerase from proceeding. So we'll say unless physical steric hindrance. Okay. This is kind of what allows cells that look different to still have similar functions. So let's just say that we have 3 different cell types here: skin cells, liver cells, brain cells. In the nucleus of all these cells, they have the same genome. So the same genes, the same DNA, the same chromosome, nothing's changing. Let's pretend right now mutations have not been acquired just by living. Okay, so there's this, here's the gene. The gene is a blue gene, but blue genes. So let's just say, stupid. Let's just say this gene is important for cell division. Okay. Yeah, that's actually a really good example. Okay, let's say that these genes, I'm addicted to blue cheese. Okay, so let's say these genes code for cell division proteins, all right? So what do we know about skin cells? Skin cells are rapidly dividing cells, right? They're always renewing. So even before looking at these cartoons, like don't even look at it. If I just asked you, do you think the genes for cell division are being transcribed? You're gonna say, yeah, because skin cells are always trying to divide and make more skin cells. And so if I said, okay, if you had to rate the rate of transcription, do you think transcription's happening at a 0 basal level, moderate level, or high level? You're gonna say high because we need these genes. Okay, great. Liver. You know the liver is important for metabolizing a lot of things, and like, you know, these liver cells, they're highly functioning, which means they, you know, with anything, you use it too much, it starts getting old and dies. So you have to replenish the liver, but maybe not as often as skin. So you would think in your head, okay, liver cells also need to replicate and divide. So, but maybe not as fast as skin cells. So let's say they're probably going to have moderate levels. No, but I chose a bad thing here. Shoot. Okay. So, okay, I know what I'll say. Okay. So then you have your brain cells. So what do we know about neurons? Neurons don't divide. So if you are thinking about it, you might say, okay, well then transcription shouldn't happen. If it happens, it's going to be very low, but those gene products won't be enough to actually allow the brain cell to divide. Okay. So let's say that you have your skin cells, and let's say that you have a damage has occurred to your liver cells. So if you think about it in that term, or in that way, you're thinking, okay, well, these liver cells, they have just been damaged. Like maybe you just chugged a 40, and now your liver cells really need to process that alcohol. So they need to make some more liver cells. Okay, so if we think about it, here's your genes, same genes in all the nuclei. And so you have this regular amount of basal transcription factors. And so skin cells are just going to be dividing at their normal rate. La, 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 we're just dividing at our normal rate because transcription's happening. No extra anything has come here. No, nothing has influenced the rate. And if you look at the liver cell, same gene, same basal transcription factors, those still come there. We have this little extra thing here. This little, this thing right here, this is an activator protein. So we just talked about activator proteins Tuesday. Alone, the activator protein does nothing. Where does this activator protein bind? It looks like it's binding to this place called a control element. Control elements is another way to say response element. It's another way to say regulatory element. So these activator proteins can bind to control elements, and when they bind there, that only when they bind there is when the magic happens. So just like we saw in prokaryotes, activator proteins, this has to be an active activator protein. So something activated this protein. Something was like, oh man, activator protein, can you please go make sure transcription happens faster? And the thing that activated this protein was the damage in the liver. So the damage excited the activator protein, and now the activator protein's like, I'm gonna go bind to that regulatory element. And when it binds there, what does it do? It recruits the basal transcription factors and or RNA polymerase at a higher rate, thus increasing the rate of transcription. So activator proteins, just like we saw in prokaryotes, increase the rate of transcription. Okay, and so neurons, brain cells, here's the brain cell in the nucleus. Same old basal transcription factors are going to bind there, but the neurons know that they really shouldn't be dividing. So there are other types of regulatory proteins called repressors or in eukaryotes, I think we like to call them transcriptional inhibitors. So these words are used interchangeably. So the repressors / inhibitors, only when active, they're going to bind their respective control elements. And in this case, here's your inhibitor bound to its respective control element, and this is going to elicit some kind of interaction between these two structures, these two, I guess, complexes. And so what ends up happening is it inhibits the RNA polymerase and the transcription factors from transcribing. Maybe a few leak out, so that's why we say very low levels. But the gist of it is that this has a big effect on the rate of transcription, thus it's much lower. So this shows you how different cell types, even though they have the same genetic material, can either turn the gene on at low levels, moderate levels, or high levels, depending on their environment, or just depending on their natural function. Okay, so when you're talking about transcriptional regulation, and I said eukaryotic cells are much more complex, they are. So it's not as easy as just getting a regulator protein onto a regulatory element. Boom, you either turn it on or turn it off. We're eukaryotes, we're complex. So other proteins can be involved too. All right, so let's take a peek at this cartoon so we know what we're looking at. Here's your DNA, here's your gene again, right? And then this time we're making sure we remember every gene has a promoter, and then inside the promoter, there's a title box. Okay, so upstream of the gene, you have these control elements, these tiny little yellow regions here. Okay. Why do we call them distal? You're like, wait a second, now there's another term I need to know? Yeah. So distal just means far away. Now people are going to say, how do you know the difference between far away and close? That I don't know. So a distal control element means far away from the gene of interest, whereas proximal is in close proximity to the gene of interest. And so these can be, both of these can be up or downstream of the gene. It doesn't always have to be upstream. It could be at the end of the gene. That's fine too. And so, yeah, just terminology. So we have these little control elements here, right? So here's your control elements. And we call these control elements enhancers. Why? Because when the activator actually binds them, it's gonna enhance transcription. So in eukaryotes, eukaryotic regulatory elements can either be called enhancers or silencers. Okay. That's the region of DNA. So remember, regulatory elements is a DNA region. If nothing is bound to the enhancer, nothing happens. Only when a regulatory protein binds to the enhancer will enhancement of transcription occur. So what binds enhancers? Activators, active activators. What binds silencers? Active inhibitors. If they are not active, they do not have the ability to bind DNA. Okay? So I'll just put this here, active, don't forget, active. Okay, so there's a little bit of terminology. Last but not least, you have the terminology cis versus trans. And so cis elements are going to be any element that is on the same chromosome as the gene of interest. That it is also nearby. So cis, let's do a different color. Cis is on the same chromosome, and by definition, well, not really by definition. If it's nearby, then by definition, it's on the same chromosome. So we call things cis regulatory elements or cis, that's really all we would say. So cis is used to describe regulatory elements. Whereas trans is used to describe elements on different chromosomes. So regulatory elements on different chromosomes. It also is used to describe regulatory proteins. So we say that regulatory proteins are actually trans proteins. I don't know why. Because I guess usually the genes that code for those proteins are on different chromosomes. So their original gene is a trans element. Okay, there is all the terminology. So now we can figure out this cartoon. Okay, so we have a gene, we've got some enhancer elements that if the activator's active, it can bind. This is just an example of enhancement. So this can also exist for silencing. So what ends up happening is somehow these activators became active and they now bind to their respective enhancer elements. And so when they bind to these enhancer elements, it actually makes the DNA bend. See how here it was this long line, and now the DNA is bending in a little U-turn. And so as the DNA is bending, well, what ends up happening is that these recruit something called DNA bending protein. So let's jot some notes down. So the activator binds to the enhancer elements. Okay, this recruits a protein called DNA, not binding, bending. DNA bending protein. Bet you can guess what it does to the DNA. Bends it. Okay, so this bends the DNA. Okay. And so when the DNA is bent, now what ends up happening is that you can see here that the enhancer elements with their activators are much closer to the promoter region than before. So see, look at this huge distance. Nothing's really going to happen there. Now you're in really close proximity. And so what ends up happening is that you have this, so we'll say it brings the enhancer complex, which contains the activators and the enhancer elements, close to the promoter. Okay, because it was distal, right? And now we have brought it so that it's nice and close. We don't call it now a proximal element. It was always a distal element, but we have taken it from very far away to very close. And so what this ends up doing is it recruits extra proteins called mediators. And these mediators, just like a mediator of an argument or conversation, it's mediating the conversation between two parties. And so this is what mediator proteins do as well. So it is basically joining, allowing the kind of like a conversation, because it's basically linking, it's acting as a bridge between these activator proteins and the promoter. It's kind of like squishing it together even more. So recruits of mediators, and now what you have is your enhancers and a big bridge of mediators, and then your promoter. Okay. So if a little tiny secret needs to be sent from the enhancer complex over to the promoter, you can. Obviously not real secrets are being sent, but what ends up happening is now you can recruit RNA polymerase to bind at with a greater affinity, and you're increasing the rate of transcription. So this increases affinity of RNA polymerase, and this thus increases the rate of transcription. Without that, RNA polymerase can still bind with the promoter, but it's going to get there and take its sweet time. And transcription will happen, but just at a very low basal rate. Okay, so, and like I said, this is just an example of activators binding to enhancers. but the same idea could be true if it was inhibitors binding to silencers. So mediators would come, and instead of increasing the affinity for RNA polymerase, they would decrease or block polymerase from proceeding. One of those two things would happen. Okay, any questions so far? Okay, cool. Let's keep going then. All right, so next is mRNA processing. So we already went through DNA. So this is chromatin assembly, right? Is the chromatin tightly wound or not? Here's your rate of transcription that we just finished talking about. And now we're going to talk about, okay, once you've made your pre-RNA transcript, we know we have to process it. And we know that it's going to become a mature mRNA. So this is something that you've seen before, but I'm bringing it up again to remind you that, that alternative splicing is actually a mechanism of gene regulation. Because it means some genes in maybe some cells, so maybe protein 1, maybe this is a gene for, I don't know, let's say like, I'm trying to think of things that would be similar but different. Okay, I know. So let's say protein 1, all 3 of these proteins, this gene is a gene that codes for structural proteins. Okay, so some cells require extra sturdy structure. Some cells need to be squishy and malleable, right? They need some structure, but not too much. They don't want to be rigid. So the cells have the same gene, so we'll call it one protein. So it's a gene that codes for a structural protein. And this gene happens to have 6 exons. Let's say in heart cells, you don't want too much structure, right? You want your heart to be able to be squishy. And then I'll just come up with one other, skin cells, right? Some rigidity, but not too much. Like you don't want to have like a Tin Man suit, but you definitely need a little bit more structure or more repellence or water repellence or whatever it may be. But you still need that same type of structure. You just need different levels of it. And so alternative splicing may allow some cells to pick and choose the exons so that the overall function of the gene is, overall function of the protein is the same, but maybe the severity of or the intensity of the function is moderated a bit. So that's really all I needed to talk to you about for alternative splicing, that it's just a mechanism of gene regulation. So alternative splicing means that in the heart cells, these 2 proteins are not made, right? So that means this mRNA is not made. That is regulating gene expression. You are stopping these types from being made, and you're only allowing for this mRNA to be made, thus translated. Okay, that's pretty much it. So let's see. Next, we have what happens with your mRNA. Like you better make sure that mRNA doesn't get degraded, and you make sure that it's stable. And then you need to actually get it to the ribosome so that it can start being translated. So we're going to start with this right here with mRNA stability. And so there's 2 things here. One is very simple, and we already know it. It's your 5' cap. And we would call this not like a very severe mechanism of gene regulation, but it's important because if you don't have that 5' cap, 2 things can happen. One, the RNA may be degraded, right? So RNases will start chewing from the 5' end and start eating that RNA. The other thing that the 5' cap does, don't forget, is it's the recognition, it's the binding site for the ribosome. If the 5' cap is there, the mRNA never gets recognized, thus never gets made into a protein, thus gene has been silenced. Okay. So the 5' cap, we would say, actually increases the rate of expression, right? Or, yeah, we don't say rate of transcription anymore because it's already been transcribed. So now we're talking about it increases the rate of protein expression or translation. Okay, so not only for protecting against degradation, but as a recognition site for the ribosome. Okay, small subunit. Okay. Now, we've briefly talked about these things called microRNAs before, several times throughout the semester, but we haven't really gotten into any extreme detail about them. So when we are talking, now we are. So microRNAs, some of the things that we've said before is that they are these short little RNA molecules. So they never become proteins and they're pretty short. So anywhere between 20 to 22 nucleotides is the average length of a microRNA. And they come in two types. They can either be 100% complementary to specific mRNAs, remember, mRNAs are the ones that get translated, or they are less than 100% complementary. Okay. And those two things, I bring it up because they're completely different mechanisms. So it's pretty interesting. So microRNAs, I would say, are these tiny little things, but over the last probably maybe 15 years, we started becoming more interested in identifying them and then learning about what they do, because we didn't really even know that they existed. And then we were like, what's this thing? Oh man, every time that this microRNA is not made properly, this major crazy thing happens. Just a change of 20 to 22 nucleotides, whether it's there or not there. This person now has neurodegenerative disease or not. Okay, we better take a bigger look at this. So in mammals, at least the last time I checked, approximately 2,200 genes in our genome code for microRNAs. So we have over 2,000 different microRNAs, which can either be perfectly complementary to messenger RNAs or kind of similar to them. Okay, and so this is in mammals. And they're highly conserved. So the same microRNAs that we have are the same microRNAs that Paloma has. She doesn't care. Okay, and then let's see. Then what else do I want to say is that, yeah, okay, so if you think about it like this in terms of our entire genome, and I know it doesn't seem like a big percentage, but this is about 1 to 5% of our entire genome, which I think is pretty crazy because these are so tiny and our genome is so giant. So I don't know. I think that microRNAs are fascinating, right? And because we can't really study them that well because they degrade really quickly too. So these microRNAs, they have the ability, so even though it's 1 to 5%, even though they're so tiny, they can, or they have been shown so far to regulate up to 30% of all of our genes. These tiny little things are what say, you can become, you can go ahead and go to the ribosome. You cannot. I'm going to destroy you. You're not going to be translated. Nope, you can. So wild. So they are very sensitive to changes in the cellular environment, and they have a very high efficacy of gene regulation. So when they do go to work, they make sure they do their job. Okay. So I think my next, yeah. Okay, good. So this cartoon is going to help me describe how microRNAs regulate gene expression via mRNA degradation. So essentially, if it's 100% complementary to a specific RNA, the mRNA gets degraded. And if it's less than 100% complementary to the specific RNA, the mRNA still stays an mRNA, but translation gets stopped. So translation gets inhibited. And this can either be translation initiation, or there's a different mechanism for stopping elongation. Okay, so let's first talk about the microRNAs that degrade mRNAs. So this, again, remember, 100% complementary to the mRNA. So this is microRNA, this is mRNA. Okay, so it's a microRNA. RNAs don't just poof, magically appear. They're coded for by the DNA. So there's genes for the microRNA. The gene will be transcribed, a microRNA is made, and then that microRNA has a function. So let's look at this. So here's your gene. Let's say that this is the region that codes for microRNA, I don't know. And when we give them name, we don't give them fancy names. We just call them MER22 or whatever number it was. MER, I don't know. But they kind of get made in the same way that an mRNA does. There's a premature, and then another premature, and then finally a mature RNA. So the way that these microRNAs are made is they have regions that are complementary to themselves. Okay? And so if we zoom in, obviously, we know that this region of the mRNA is complementary to itself. So if you think about it, let me try to draw this. So I'm going to try to draw it before it folded. Okay. Looks like, just to kind of keep it color coded for everybody here. So this was 5' end, this was the 3' end. And so these are the regions, I'll just highlight them in yellow. Those are the regions that were complementary to one another. Can't really see, so what ends up happening is that mRNA folds back on itself and forms a hairpin loop. So this first one is called the pre-miRNA. And then once this happens, a molecule called drosha, so this is a protein. It is a protein that has exonuclease activity. So drosha has the ability to exonuclease the ends of this microRNA. So really, if you have 5' and 3', right? So it starts gobbling both ends up and then starts until it reaches this hairpin stem, right? So this region is now gone. It was removed by drosha. And so this pre-PRE microRNA then gets modified more or processed more by another molecule called dicer. And so what dicer does is dicer has helicase activity. So this is exonuclease activity. This is helicase activity. And we know what helicases do. We go in here. Okay. Dicer is going to cut those hydrolyze those hydrogen bonds. And so what you're left with is just the mature microRNA. So it's single-stranded. And so this is the thing that's 20 to 22 nucleotides long, the mature microRNA. The pre and the pre-1 and pre-2 are much long, not much, but they're longer than that, right? But they kind of get chewed away and degraded. So this right here is what is 100% complementary to a piece of mRNA, not the whole thing because mRNAs are long. And ooh, my apologies here. So this should say AGO2, and it's a complex. So it's called Argonaut. So it's Argonaut 2, and it will bind to the microRNA. So when Argonaut 2 binds to the microRNA, so this right here is called a microRNA complex. So the Argonaut, you can see here that it was also a microribonucleic protein. So in that Argonaut protein, there's also RNAs involved. So you have this Argonaut, so this is your go-to / microRNA. This is what allows this complex to bind to the specific region of the mRNA. So here it is bound to the mRNA, and then this marks the mRNA for degradation. So RNases will come and degrade that entire RNA. And that's it. And so now the mRNA cannot go to the ribosome, it cannot be translated because it doesn't exist. And that's pretty much it. So yeah, and it really starts with, the degradation really starts with Argonaut. So this complex, what it ends up doing is once it binds to the RNA, so let's say binds to the mRNA. Once it binds, the Argonaut starts to degrade the duplex. And by duplex, I'm talking about the micro to the mRNA duplex. So it will start chewing up this specific region. And then once you've cut a big hole in the mRNA, then other RNases come and finish up the job. Okay. Alrighty. So that is microRNAs that are 100% complementary to messenger RNAs. Okay. So these degrade the entire mRNA. I think we can let's go ahead and talk about the other type of mRNA, and then I'll ask if there's some questions. Okay, so now mRNAs can definitely make sure that the microRNAs can definitely ensure that the mRNA is stable, but microRNAs can also make sure or not that translation happens or doesn't. So let's talk about microRNAs that can either inhibit or stop initiation or elongation. So I think you can see in general that microRNAs tend to be associated with decreasing or stopping gene expression. Okay, okay. So we'll talk about it 1 second because I just have one quick thing to say about IFs. Remember what IFs are. These are initiation factors, and they are machinery for gene regulation. So we would say that IFs, when they're activated and they do their job, they increase expression, right? Because you can't start translation unless the initiation factors are there. And guess what? The initiation factors aren't just floating around waiting to do their job. They have to be activated. If they are not activated, then they don't start translation. So a mechanism of gene regulation is actually IF activation. So here is another mechanism, is IF activation. And these IFs, here's an example. You know how most times people are like, oh yeah, phosphorylation usually activates things. And I was like, true, but there's always some times that phosphorylation doesn't. So IFs that are inactive are phosphorylated. So to become active, they need to be dephosphorylated. So dephosphorylated IF is active. Okay. Not the only exception, but yeah, I would agree. Phosphorylation is typically associated with activation, but initiation factors of translation are an example of when dephosphorylation is absolutely necessary for activation. So that means that activation requires phosphatases. Right? Because phosphatases remove phosphate groups. Okay, cool. That's all I wanted to say about this. So now we can move on to microRNAs. Okay, so translation can be inhibited by microRNA. And so the way that you would make the mature mRNA, microRNA is exactly the same. So we just talked about this. Totally the same. But the difference here is that this microRNA is not 100% complementary to a specific mRNA. It's maybe like 80%, I don't know. So the same thing happens though. A complex of microribonucleoproteins recognize the microRNA and they bind to it. So we just finished talking about what happens if it's 100% complementary. The complex is called Argonaut 2. Now, how does the cell know whether it's gonna be complementary to an mRNA or not? These are questions I also have. If it's partially complementary, a different Argonaut complex, and I have just number there because it differs per mammal. So some use Argonaut 1, some use Argonaut 4 and 5. So just know it's a different complex than when the microRNA is 100% complementary. So a different complex binds to the microRNA. It still allows this to bind to the microRNA, but you can see here is the region that's not complementary. And that's why no hydrogen bonds are formed there, that little region. And so usually what happens is that because it's not perfectly complementary, this complex, multiple complexes can bind along the mRNA. So this is one complex, this is another complex, and they can bind throughout different regions of the mRNA. So how do we stop initiation or elongation of translation from happening? So let's take a peek at it. All we have here in this slide is that translation is inhibited. What do you do? How? So here we go. Here is inhibition of initiation. So let's start there. Okay. So what ends up happening, let's describe the cartoon so we know what's happening. So here's your 5' cap. Right? And we know that sometimes there's a lot of nucleotides between the 5' cap and the start codon. So this is the 5' untranslated region. Put it in red so we remember. So, oh, it's in red. It's right there. Okay, here it is. All right, and then the ribosome. So the ribosome has to recognize this. It binds to this. So let me try to draw like 5' cap, and then we have RAG, and then we have, I don't know, CGA, blah, blah, blah. Okay, so when we talked about transcription, we know that the first thing that has to happen is that the small subunit is going to bind around the 5' cap. It recognizes that thing. So that is assuming that the 5' cap is accessible, right? So here's your small subunit. Small subunit. So this relies on the fact that the 5' cap is accessible to translational machinery. Okay, so what ends up happening when a microRNA in its big giant complex is formed, I am going to modify this from here so it's not going to confuse you. We're just going to call it some number. Argonaut complex. You can see not perfectly complementary. What ends up happening is that these complexes tend to favor, so we'll call it the Argonaut microRNA complex, they tend to favor 3' UTR regions. I don't know why. So they bind there. And what else is interesting is that the 5' cap, it must be something about the Argonaut complex. It has an affinity to methyl. I'm not sure why, but the 5' cap has a high affinity for the Argonaut microRNA complex. So it binds there. In doing so, now the 5' cap is inaccessible. Here it is. tied up in some other business. So if the 5' cap is inaccessible, the ribosome can't find it. So this causes small subunit cannot bind. If the small subunit cannot bind, translation never starts. Easy as pie. Okay. So you see your subunits are like, oh, please let me come in. Nope, sorry, busy. Here's your initiation factor that's been dephosphorylated. Oh, let me help. Nope, not gonna happen. The 5' cap is now inaccessible because it bound to the microRNA Argonaut complex, and some people call it the RISC complex. So that's what this means. The micro RISC complex is just indicating Argonaut with the microRNAs. Okay. All right, so let's see how elongation is blocked. Let's say the 5' cap was still accessible, the small subunit bound, and translation is starting and it's happening. So this is going to be how we stop elongation. Okay, so what are these structures in this funny cartoon? So here's your small subunit, here's your large subunit. Here is, I don't know what this is. I'll think about it in a second. And here is your mRNA, right? So here's the 5' cap, the small subunit already bound there, already started sliding down. Oh, this is the amino acid chain that's being made. Okay, so here's your N-terminus. This right here is your methionine, and then you have your next amino acid, et cetera, et cetera, et cetera. Okay, so this shows you that that translation has already started. So the ribosome's moving along its merry way, but you have to remember that downstream, there is a RISC complex bound. So here's your RISC complex, here's your microRNA that's not perfectly complementary. So here's the region that is not complementary, not complementary. Okay. And then it's kind of towards the 3' end because we say that this tends to be in the 3' region. Okay, maybe it's not all the way in the 3' untranslated region, but it's kind of downstream of the start codon. So what ends up happening is the ribosome, once it gets here, it can't move because there's a giant RISC complex in the way. So a couple things is that there is a steric hindrance, right? And so just like polymerases, if the ribosome stalls for too long, it will start to disassociate. And that's what ends up happening is that the ribosome disassociates, and then you have this small chunk of amino acid that was made, and typically that's not going to have a function. So it's going to be this truncated version of the protein, but the protein wasn't made. And so you can see that like there are certain times in a cell's life or in a cell's day, whatever we're talking about here, that, you know, it originally made an mRNA, but then something happened where it's like, oh, we need to divert our energy. We actually don't need to make that into a protein right now. And so the way that they respond to it is they actually quickly transcribe the microRNA. The microRNA binds to the Argonaut complex and then stops translation or degrades the mRNA. And so given that something that happens normally, you can also see if microRNAs are misbehaving or are, you know, starting to be overexpressed and overmade, they're going to start messing with gene regulation in a bad way. So microRNAs, there is actually a huge correlation between microRNAs and neurodegenerative diseases and also cancer. So like I said, it was a huge boom in the research industry. Like we wanted to know all the microRNAs possible. And like good scientists would do, we were like, wow, we can just get this tiny little RNA piece and we can stop translation from happening, or we can degrade RNA. Cool. How can we make this happen in the lab? And so in the lab, we actually can do this. And in the lab, we call them short hairpin RNAs. Or we also call them silencer RNAs, so siRNAs. And so we can manipulate or just create these short little RNAs to target our gene of interest and make sure that gene of interest gets degraded. So for some reason, maybe a scientist wants transcription to happen, but then they don't want to interfere with transcription. They're like, we don't want people to say like, oh, is it because transcription didn't happen? So we're going to say, all right, let transcription happen, but then we're going to stop the mRNA from being translated. And so these people, or the people that came up with this in the lab, are Craig Mello and Andrew. I may have switched their names. Andrew Mello? No, Craig Mello and Andrew Fire. So these are two scientists that used C. elegans they actually discovered microRNAs in C. elegans, and then were like, oh my gosh, we can use this to silence genes. And so they came up with this short interfering RNA or silencing RNA, and they ended up winning a Nobel Prize. And this was not very long ago, maybe 20, 30 years ago, maybe. So I think this is really awesome. And this was for CRISPR. This was like a really cheap and dirty way to repress or inhibit or degrade your transcripts. Fire. Okay, and then excuse me. Last but not least, we're going to hit this last little asterisk of how you can regulate genes. And so we have to remember that proteins, even if they're folded even when they're made out of the ribosome, they're in their primary structure. So they have to get folded properly. And then sometimes they even have to get modified with additional functional groups. So I'm just reminding everyone that modifications of proteins are in fact a mechanism of regulation. So the last mechanism is the way a cell modifies proteins and whether it does or not. So if a protein needs to be phosphorylated, a kinase needs to do that. So that kinase has to be activated. And what activates the kinase? Something else or some kind of cue. And so don't forget that some enzymes are responsible for modifying proteins, but they have to be active. If the enzymes are inactive, they're not going to modify the proteins. If they don't modify the proteins don't function. So activating enzymes is a regulatory mechanism. And so it gets even more meta. Some proteins themselves first need to be modified before they can go modify other proteins. So maybe an kinase needs to be active. In order for it to be active, it needs to be phosphorylated. So it needs to be modified. This is what makes the kinase active. Now this active kinase can go activate another protein by modifying it. Okay. So we'll say it modifies the target. So if you're modifying the target, you are activating the next gene or protein. And this can continue. It's kind of like the signal transduction thing. And then last but not least, one last mechanism of at the level of the protein is that sometimes proteins, they need to be marked for degradation. And so degradation is also a modification. So proteins can be degraded when they were marked with ubiquitin or simulation or sumo functional groups. So that is another modification. And like the IF, if you want to stop it from doing its job, phosphorylate it. Some other proteins, if you want to stop them from doing their job, the only way to do that is to mark them with ubiquitin and degrade them. Okay, so the modification here is ubiquitin. Okay, and I think that that covers it. So now we're all full circle, right? So once you have modified these proteins and folded them the correct way, now these proteins can go do what they're supposed to do, whether that's replicate DNA, whether that's transcribe, whether that's process, whether that's translate, whatever it may be. And that's it. So any questions? Okay, so I see question marks here. Max, did your question get answered? Because I don't know when that was or what that was in reference to. Yeah, okay, cool. Alrighty then, that is it. So that covers our discussions on gene regulation. And that's just scratching the surface, but it's actually really, really important to disease research. Usually diseases are the product of genes being misregulated, and a lot of other diseases are because of gene mutations, but misregulation usually occurs because a product was not made properly. Okay, so next week we are starting with, I forgot. Oh yeah, cell cycle. So I know everyone's like, I've done mitosis 100 bajillion times, like I get it. And I'm telling you, you don't get it at the level that we're gonna do it at. So review what you do know about the cell cycle, review what you do know about mitosis, but we are gonna talk about regulation of mitosis. We are gonna talk about like what specific proteins are actually pulling the chromosomes apart and how. And so I think that we're going to go into a lot more detail. So do yourself a favor, just at least look at some of the resource videos that I post for you, that I will post for you in the next day or two. And that's it. So have a great day. If you're in my 10 o'clock class for lab, come prepared. My 10 o'clock meeting, come prepared. These have been really fun to listen to. I'm excited to hear more. And I will see everyone else later. Have a great weekend, and that's it.